Hello students, welcome to Bachelor of Science program, subject Botany, semester 5, course code BOD105, course title Nursery and Gardening. The title of this unit is Seed and the module name here is Structure and Types of Seed. I am Ms. Aditi Venkatesh Naik, Assistant Professor from PS College of Arts and Science, Farmagudi. Outline, you'll be studying the structure of seeds and type of seeds. What you will learn from this course, you'll be able to state the types of seeds and differentiate between the structure of monocot and dicot seeds. What is a seed? Student, seed is the basic organ of a plant. There are various definitions for seed, and seed is defined as the matured and ripened ovule which is formed after fertilization and with the tissues from the mother plant. The formation of a seed is nothing but a process of reproduction in the crop plants. The seeds contain reserve materials like starch, proteins, which are the primary metabolites, along with other components which could be secondary metabolites and are necessary for proper embryonic development and growth of plants. The seed, as we all know, is found inside the fruit which converts into a new plant when we plant it. Hence, the seed is a very important plant organ. When we look at seeds, they possess immense diversity in the internal and external structure. These differences are to an appreciable extent related to the dispersal and germination strategies. It also involves the size and positioning of the endosperm and embryo, the structure, the texture, the color of the seed coat, and the shape and dimensions of the seed as a whole. When we look at seeds, as I already said, there are varying sizes of the seeds. The minute seeds of orchids weigh as little as 2 into 10 raised to minus 6 gram each. So you can understand how tiny the seed is and how much of weight it carries. Whereas the seeds of legume like Mora oleifera are possibly the heaviest and the weight of each exceeds 1000 gram. Whereas one seeded fruit of that of palm like Lopodio, Lodiocio maldivica weighs more than 20,000 gram. The seeds basically consist of three main components, that is the embryo, the endosperm, which is sometimes referred to as the perisperm, and the seed coat. Both the endosperm and the embryos are the products of double fertilization, whereas the seed coat develops from the maternal ovular tissues. As I already said, the structure of the seeds is all dependent on the number of cotyledons that are present in the seed, and accordingly, the seeds can be categorized as monocot seeds and dicot seeds. When we look at monocot seeds, the monocot seeds are seeds which bear only one cotyledon. Example, paddy, wheat, maize, ragi, bajra, sorghum, banana, and so on. Whereas the dicotyledonous seeds are seeds which have two cotyledons. Examples are chickpea, bitter gourd, castor, black grams, horse grams, tobacco, lettuce, mango, neem, bottle gourd, and so on. Now let us look at the structure of the monocot seed. When we look at the structure of the monocot seed, we need to recollect the seeds which belong to the monocotyledonous plants. Now these monocots bear only one cotyledon, and as you can see here, this image shows the structure of that of the maize, and it comprises of the seed coat, the endosperm, the aileron layer, the embryo, the scutellum, the embryonal axis, coleoptile and coleopryza. When we look at the seed coat, it is in most of the seeds of cereals such as maize, the seed coat is membranous. You can recollect how the maize seed looks, the corn seed looks. It has membranous layer and it is generally fused with the fruit wall called the hull. The endosperm is nothing but a bulky substance and stores food. Generally, the monocotyledonous seeds are endospermic, but sometimes, in case of orchids, the seeds may be non-endospermic. Here in this particular image, you can see the yellow portion or the orange portion resembles that of the endosperm, which is the nutritive tissue. The aileron layer is the outer covering of the endosperm, which separates the embryo by a proteinaceous layer called the aileron layer. And the embryo is the crucial part of that of the seed, which is small and is situated at the groove at the end of the endosperm. Now, this embryo comprises of various organs or various parts. Whereas when we look at the scutellum, okay, it is one large and shield-shaped cotyledon. The embryonal axis consists of the plumule and the radicle and form the two ends of the embryo. Whereas the coleoptile and coleophysa are nothing but the plumule and radicle which are enclosed in sheets, and these are the coleoptile and coleophysa which are capable of giving rise to the shoot and the root respectively. Now let us look at the structure of the dicot seed. The dicot seeds are made up of two cotyledons, and as you can see in the image, same as that of the monocots except the number of cotyledons, most of the plant parts or the organ parts remain the same. That is the seed coat. Here there is hilum, a micropyle, embryo, cotyledons, radicle and plumule and endosperm. Now let us look at each of these parts. The seed coat, as we all know, is the outermost covering of the seed. The seed has two layers here, the outer which is known as the testa and the inner which is referred to as the tegmen. The hilum is nothing but a scar which is formed on the seed coat through which the seed was initially attached to the fruit, or the developing seed ear was attached to the fruit. The next is the micropyle, which is a small pore which is present just above the hilum. The embryo, which is again a significant part of the seed, comprises of the embryonal axis and two cotyledons. The cotyledons here are often fleshy and full of reserve food material. Again, the embryo comprises of radicle and plumule, which are present at the two ends of the embryonal axis. The endosperm in some seeds of this particular seed, that is dicotyledons such as castor, the endosperm formed as a result of double fertilization and is the food storing tissue. Here you can see the seed which is cut open, and here you can see there is development of embryo that is taking place, which has a plumule and a radicle. In plants such as bean, gram and pea, the endosperm is not only present in the mature seed, they are known as non-endospermous. As already said, the type of seeds are of two types, that is monocotyledonous seeds and dicotyledonous seeds, which are basically based on the number of cotyledons. Monocotyledonous seeds are seeds which have one cotyledon, example like orchids, corn, rice, etc. Whereas dicotyledonous seeds are the seeds that have two cotyledons, example gram, pea, bean, mustard, mango, and so on. Now, based on the presence and absence of endosperm, the seeds can be further classified as albuminous seeds and exalbuminous seeds. The albuminous seeds are also referred to as endospermic seeds. As the name suggests, these are the seeds which have the presence of endosperm. In albuminous seeds, here the cotyledons are thin and membranous, and endosperm persists and nourishes the seedling during its early development. Example, castor, poppy, custard apple, and so on. The next group is the exalbuminous seeds, which are referred to as non-endospermic. Here the seeds lack endosperm and are called non-endospermic or exalbuminous seeds. In exalbuminous seeds, food accumulates in the endosperm tissue at an early stage of development, but it is utilized by the developing embryo and the mature seeds without endosperm. In such seeds, the cotyledons store food and become thick and fleshy. Example, gram, pea, bean, etc. As we have already seen, the seeds are of the two types, dicotyledonous, seeds which have two cotyledons, and monocotyledons, that is seeds which have one cotyledon. The dicotyledonous seeds can be classified as X-albuminous or non-endospermic seeds, and they can be classified as albuminous seeds or endospermic dicotyledonous seed. Whereas the monocotyledons, based on the presence or absence of endosperm, can be classified as albuminous seeds which have endosperm called as albuminous, monocotyledonous albuminous seeds, and monocotyledonous X-albuminous seeds are the seeds which lack the endosperm, like orchids. Let us summarize what we have learned. We have seen that a seed is the basic part of the plant and is formed by the fertilization. That is, they are nothing but ovules after fertilization which develop into seeds. The seed here has three parts, which are the seed coat, the endosperm, and embryo, where the embryo is the most significant part of the seed, which comprises the epicotyle, hypocotyle, plumule, radicle, and cotyledons. The seeds can be broadly classified as monocotyledonous seeds and dicotyledonous seeds, which are further classified as endospermic or albuminous seeds and non-endospermic seeds or X-albuminous seeds based on the presence or absence of endosperm. These are few references that you can refer. Thank you so much.